So hello, everyone. My name is Maxime HERVE. I'm working at the Institute for Genetics, Environment and Plant Protection of Rennes in France. And on behalf of myself and Anne-Marie Cortesero, I will do this presentation on the development of insect-resistant oilseed rape cultivars. I thank the organizers for inviting us to do this presentation, which is in fact an update of a previous presentation I made 4 years ago for a previous GCIS meeting. So as you all know, oilseed rape, and especially winter oilseed rape, has a long cycle over several seasons and is threatened by many different insect pests attacking at different stages of the plant. And among these pests, you can find, for example, beetles, weevils, or flies and midges or caterpillars and so on. There are many different species which depend on the season and also on the country. These insect pests are mostly controlled using insecticides, especially pyrethroids, but you know that resistance to pyrethroids are spreading in insect populations. And here you have two examples from German data on the pollen beetle and on the cabbage stem flea beetle. And you can see here that over 10 years, from 2005 to 2015, there was a dramatic increase in the proportion of populations of the pollen beetle resistant or even highly resistant to pyrethroids in Germany. And here you have a screenshot of the situation 5 years ago in Germany. All the stars in reds are populations of the cabbage stem flea beetle that were already resistant to pyrethroids. So we have a big problem with the control of these insect pests, you know that, and one of the alternative solutions to insecticides is breeding for insect resistant cultivars. This is a strategy that is quite common in other crop species like maize or wheat, for example, but today there is no resistant oilseed rape yet on the market. So one of the main questions is why? And we think that in fact, the main problem that impair the development of these insect-resistant cultivars is phenotyping. I will not develop these points I developed in the previous presentation a few years ago, but to summarize these, the process of phenotyping, and especially large-scale phenotyping, is very difficult in general with insects, but especially with insects that are pests of oilseed rape. And anyone who engage himself in phenotyping, large-scale phenotyping for insect resistance in oilseed rape knows that it really it's a bottleneck in the process of breeding programs, and even identifying sources of resistance studies are limited because of this challenge of phenotyping. So this probably explains why not so many studies were performed on insect resistance, but still quite a number of studies have been published on this subject, and I will try to summarize the results of these studies in this presentation. Broadly speaking, there were 3 main strategies that were explored to make oilseed rape resistant to insects. The first one is the introduction of transgenes into the oilseed rape genome. The second one is the selection for resistance already present in some oilseed rape cultivars. And the third one is the integration of resistance from other Brassicaceae species, which could be crossed with oilseed rape. So I will summarize on the 3 different strategies what has been done so far. Starting with the transgenes, of course, or at least I think, of course, the first attempt that has been done is to produce Bt oilseed rape. So oilseed rape expressing toxins coming from a well-known bacteria, and quite a huge work has been done on this. And here you can see that this Bt oilseed rape was really effective against some caterpillars it targeted. The damage was completely null on the Bt oilseed rape compared to the control oilseed rape. But as you may know, resistance to Bt toxins in insect populations are spreading very quickly, and in fact, as quickly as the resistance to insecticides. So to my knowledge, there is no Bt oilseed rape so far, and I'm not even sure that research is still going on this topic. Apart from these toxins coming from bacteria, there has been some work on proteins that disrupt the digestive system of the insects, and in fact on 3 different classes of proteins: protease inhibitors, lectins, and chitinases. So starting with protease inhibitors, some work has been done targeting some coleopteran species, so weevils and the cabbage stem flea beetle, and also in parallel some work on lepidopteran species. Here it's the caterpillar of the diamondback moth. And to summarize the results of this study, in fact, the problem is that this strategy is not really effective because insects already possess mechanisms circumventing the negative effect of protease inhibitors. Here on this figure from one of these papers, which was on the cabbage seed weevil, you can see results on 2 different strains of the weevil. Here on these strains, you can see that the performance of the weevil, so the efficacy of the transgene, was not higher on the transgenic oilseed rape compared to the control oilseed rape. And even worse, with this strain, the performance of the insect was even better on the transgenic oilseed rape compared to the control oilseed rape. And in fact, it's because insects already possess mechanisms to circumvent this effect. And in fact, when there is some protease inhibitor in the food, other enzymes, digestive enzymes, are increased in their quantity. And so insects can even take an advantage from this food. So in fact, the performance is better in the end. The second strategy is lectins. So it's also proteins that impair the digestive system of the insects. Quite a number of studies has been done on the pollen beetle. And here you have one of the first results where you can see that the survival of the beetle was much lower on food where lectins were added, lectins coming from different plant species, compared to control food without lectins. So following these promising results, transgenic oilseed rape producing P-lectin in the pollen has been produced, but the results which were obtained in the first step in the lab were not confirmed in the greenhouse. So there was even no experiment in the field, and this research finally was abandoned. And to finish with the digestive system, with chitinases. Chitinases are enzymes which degrade the insect cuticle, so therefore also the gut epidermis, which basically is cuticle. There were 2 papers published which combine a transgene producing a chitinase to another transgenes. In the second study, the second transgene was producing a protease inhibitor. And in the first study, quite exotic, the second transgene was producing a toxin isolated from the venom of a scorpion. And in both cases, it was effective against some caterpillars. You have here an example where you can see that the survival of the insect is much, much lower on the transgenic oilseed rape compared to the control oilseed rape where the survival is 100%. But to my knowledge, nothing has been published since these 2 studies, so I'm not sure research is still going on this topic. And the last example on transgenes is not about toxins or chemicals in general, but it is about physical properties of the plant. And it's the quite famous example of hairy canola. Hairy canola is a canola, so oilseed rape, which trichome density on the cotyledons and the first leaves, so it's on young plants, was dramatically increased compared to normal oilseed rape. And this was possible by transferring to the oilseed rape genome a regulator gene from Arabidopsis that regulates the production of trichomes. And these were quite effective on flea beetles and on some caterpillars. Here's a result on flea beetles. you can see that, for example, here, compared to a control oilseed rape, which was not hairy, the hairy oilseed rape, or hairy canola, it was in Canada, has suffered damage from flea beetles that were reduced to the same level as here a control oilseed rape with insecticides applied. So it was as effective as insecticides. And the same for cotyledons and the first, second, and the third and fourth leaves. So on all the young plants, this was effective. And the same result was obtained with this caterpillar, the diamondback moth. There is no treatment with insecticides here, but you can see that compared to the control oilseed rape, the hairy oilseed rape suffer less damage from this species, which is a pest in Canada. But to my knowledge, this has not led to commercialized hairy canola. So I would like to know more about that, but to my knowledge, there is maybe not so many studies continuing on this. So this is it for transgenes. So some attempts, but to my knowledge, nothing really continuing right now, but I could be wrong, especially with the hairy canola. So the second strategy, which is of course the most natural for breeders, is to look for resistance already present in oilseed rape, so in some oilseed rape cultivars. And quite surprisingly, not so many studies were published on this strategy. There was not so many phenotyping experiments that were published. And I think it could be because negative results are not always published, but it could also be because this kind of phenotyping is, as I said before, a big challenge and very difficult to set up and to follow, especially in the field. But still, there has been some studies. Two studies from German colleagues on the cabbage stem weevil showed that there was some limited but still variation in the number of larvae, so let's say the damage, between different oilseed rape cultivars. So limited variability, but still some existing. And the same was found by the same colleague, in fact, on the rape stem weevil. So different species, but attacking the same organs. Here you can see let's say it's damage here. You can see some variability, but quite limited. And what is interesting is that maybe the only accession that could be considered consistent is not an oilseed rape cultivar, but it's a resynthesized oilseed rape. And I think this is an important point to mention, and I will come back to this later in the presentation. On the pollen beetle, more studies have been published, including studies from our lab. And this is from one of our first screenings on different oilseed rape genotypes or cultivars. There was some variation for the level of damage suffered by these plants, but in fact, now we know that there is nothing you can really call resistance here. This one is just less susceptible, but it is still highly susceptible, and it cannot be considered resistant. So there is as far as I know, there is no resistance to the pollen beetle in oilseed rape. And maybe more hope, with more hope, there was one or two studies published on the cabbage aphid and the cabbage root fly, who found cultivars that were considered resistant to these species. So only 2 for the cabbage root fly, but still 2, and a bit more for the cabbage aphid. And what was maybe interesting there is that 2 cultivars here were the same as the one considered resistant to the cabbage root fly. So I don't know if breeding programs, or at least continuing research, is done on these cultivars, but maybe some hope here to find resistance in oilseed rape. And basically, that's it for this strategy. There has, to my knowledge, nothing published except the studies you have here in front of you. So the very few studies. And just to say a word about the cabbage stem flea beetle, which is one of the main pests of oilseed rape in Europe, especially in Western Europe. It's a big problem. There is no study published on the cabbage stem flea beetle, but I know some labs are working, are phenotyping oilseed rape cultivars. We are doing, or at least we were doing, because from my perspective, I can say that even if nothing is published, there is maybe not so much to hope within oilseed rape. So resistance should maybe be looked for elsewhere than in oilseed rape. And this is where we come to other species, other brassica species. You can see that not so much studies have been done, or at least published, looking for resistance within oilseed rape, but many more studies were performed looking for resistance out of oilseed rape in other brassicaceous species that could then be crossed with oilseed rape. So this is the third strategy, introgression of the resistance from other species to oilseed rape. And maybe the most beautiful example that exists in the literature is from Canadian colleagues who worked a lot on the cabbage seed weevil. In many studies, they screened for different brassicaceous species, and they repeatedly identified one species which was really resistant, highly resistant to this insect, is the white mustard, Sinapis alba. And these Canadian colleagues, they crossed the white mustard with oilseed rape, and they succeeded in introgressing the resistance of Sinapis alba into elite cultivar of oilseed rape. And even better, you can see here that the level of damage, you can see there, suffered from the plant on different genotypes obtained from the crossing, the breeding process. This level of damage was negatively correlated to the quantity of a metabolite, which was further identified as a flavonol glycoside, which is expressed in the inflorescence bract. And this is really, really interesting, first because understanding the mechanisms of resistance is an interesting task, but also because if I come back to the big problem of phenotyping, one way to avoid the large phenotypings that need insect is to have to identify some biomarker metabolites or even molecular markers, and then being able to do phenotyping based on these markers or metabolites instead of the insects themselves. So this is really a really nice example because they succeeded in introgressing the resistance into oilseed rape, but they also allowed to do biomarker-assisted selection, which is maybe one of the most promising ways to develop insect-resistant oilseed rape. And the same Canadian colleague, they also did a very nice work on the cabbage wood fly, which is quite a big pest of canola in Canada. And again, they started by looking for resistance in other brassicaceous species. And again, they found the white mustard resistant to this insect. And again, they succeeded in introgressing this resistant into oilseed rape. So they produced oilseed rape that was resistant to this insect species. I'm not really sure that this research is continuing. Maybe Canadian colleagues can answer this question in the discussion, but at least it was a very exciting example of what is possible. And parallel to these studies on this insect by Canadian colleagues, other colleagues in Europe screened for resistance into other brassicaceous species, and they identify another species which could be promising, which is a wild brassica species. It's Brassica fruticulosa. So maybe it's a species to retain, even if it's maybe less easy to work on this species compared to the white mustard that is cultivated. And if I follow up with the white mustard, which is a species that I really like, it was also identified as a resistant species to flea beetles, again in Canada. Whatever the choice or no choice test, you can see here that the damage was lower on the white mustard than on oilseed rape. So this species is also resistant to the flea beetle. And to continue with the cabbage stem flea beetles, recent work from German colleagues showed that Sinapis alba, again the white mustard, was the most resistant species they tested against this insect. Here you have the difference in performance between each of these species, and the reference being an oilseed-rape cultivar. Here you can see that the performance of the insect is lower, much lower on the white mustard than on the control oilseed rape. So the white mustard is also resistant to this species. And the same result was found with the pollen beetle. Here you have the number of larvae of the pollen beetle per plant, but you could say the same with the feeding damage. The feeding damage was much lower on the white mustard compared to the oilseed rape. So other species were identified. This is just an example, but again, I would like to focus on the white mustard because I think you guessed that I find this species really, really promising as a source of resistance. And to finish, the last example is from the same German colleagues here, a study on the cabbage stem weevil. They tested the white mustard, but sadly, it was not resistant to this species. It's not resistant to everything, but other Brassicaceae species, which are wild species, were resistant to this insect. Here you can say it's number of larvae per plant, let's say damage. Damage was much lower on this species compared to the control oilseed rape cultivar. So there is also some resistance, at least to this species, in these quite unusual Brassica species. So to summarize all of these results, is it a dream to breed oilseed rape that is resistant to insect, at least some insect pests? Well, for sure, no, it's not a dream, because some very nice studies already show that it is possible. It is possible to find some sources of resistance and to import this resistance into oilseed rape. But my conclusion from all these studies would be that this will probably be very difficult based on existing oilseed rape material, because as you have seen, there is not so much resistance within oilseed rape. So we need to find resistance elsewhere than oilseed rape. And I interpret this lack or at least low levels of resistance in oilseed rape in light of the very low genetic diversity we know in these species. So my main message is that if we want to develop insect-resistant oilseed rape cultivars, we need more diversity. And I think you understood that what I consider as the most promising source of resistance is other species, other Brassicaceae species, and especially the white mustard. I made a focus on the white mustard. It's not the only other species which has been identified, but it's striking that it's a species that was found resistant to many different insect pests of oilseed rape. So I don't say that it's not magic. We could not introgress just into one cultivar the resistance to many insect species, but at least these species, the white mustard, should be studied in depth to understand why is it resistant, what regions of the genome are responsible for these resistances, and to try at least to introgress multiple resistances into oilseed rape from these species. And a point I mentioned earlier is that another source of diversity is also resynthesized oilseed rape. And for sure, this will be much easier to cross with the actual cultivars than the other species. And there was at least one or two studies, especially from colleagues in Germany, that has shown that at least some resynthesized lines were more promising than the current oilseed rape cultivars. And to finish on the diversity, I would like to say that looking for diversity to find resistance is fine, but there is also another level of diversity which will be for sure, or I think I am convinced, will be really interesting to develop. It's the within field diversity. I mean that it's well known that when you combine at least several genotypes of the same plant, you increase the mean resistance of the plant community. This is called associational resistance. And this is something we don't do so much with oilseed rape, but maybe it would be also interesting to investigate pools of, I don't know, 2 or 3 genotypes that together are resistant to insects, rather than trying to develop one cultivar that is resistant to many insect species. And we can even go further with diversity. It will probably be very interesting and maybe successful to develop the oilseed crop diversity. So not only canola or oilseed rape in Europe, but other species. Some of them were cultivated before, but are not so much yet. And this is another level of diversity. And for sure, from all the ecological literature, that would be probably beneficial to control insect pests. I insisted quite a lot on the challenge of phenotyping. So again, I think biomarker assisted selection is a promising way to be able to perform large scale phenotyping screenings that are, of course, needed for the breeding programs. And the final word I would like to say is that we are focusing here on resistance, but we should not forget that, of course, resistance is not breeding, is not the only lever that we could use to protect crops against insects, but breeding for resistance is one thing, but maybe something which could be envisaged and maybe explored is breeding for tolerance instead, or added to resistance. Tolerance is the ability of the plant to recover from the damage. And we know, for example, with the pollen beetle, that even if there is a lot of pollen beetles on the plant, and even if the plant suffers a lot of damage, if it's in good health, it can tolerate this damage. And in the end, the impact on the yield is nothing. And in some situations, the yield could even be better on plants that were damaged by the pollen beetle than on plants that were not damaged. So, of course, it's a bit more difficult to work on tolerance than resistance, but I think this is also a thing we should keep in mind that could maybe be explored. Then resistance is not everything, and maybe tolerance will be in some way easier to manage in the field. So with these take-home messages, I will end my presentation. Thank you very much for your attention. Again, thanks to the organizers for inviting us to prepare this presentation. I will not be there for the questions, but if you have some questions, prepare them, and Anne-Marie will be there online to discuss with you. So I encourage you to discuss with Anne-Marie about all these exciting research. Thank you very much.